Okay, so today I'll be telling you about polyploid gene expression on two timescales. And I'll start with an introduction to polyploidy. So polyploidy comes in a couple different flavors, and I'll be telling you about allopolyploidy, which is formally defined as a merging of structurally different chromosome sets accompanied by genome doubling. More informally, we tend to think of this as two different species hybridizing to produce an offspring that contains the complete genome of both of its parents. This process requires at least one unreduced gamete, that is, a gamete in which the chromosomes failed to properly segregate during meiosis. And you might think that it's super rare, and in mammals it is, but actually in plants, polyploidy is quite common. So around 50 or 70%, depending on how you count it, of angiosperms are expected to be polyploid, and more than 90% of ferns are polyploid. So it's really quite common in the plant kingdom. And one of the terms that gets thrown around a lot in the field of polyploidy is this idea of genome shock, which is a term that was inadvertently coined by Barbara McClintock in her discussions of transposons. She said, "My experiment involved challenges which the genome is unprepared to meet in an orderly, programmed manner. Our present knowledge would suggest that these reorganizations originated from some shock that forced the genome to restructure itself in order to overcome a threat to its survival." And so people took this idea and applied it to polyploids because polyploidy, unlike other kinds of responses like heat shock, for example, is not a really predictable response. Different polyploids seem to respond to polyploidization differently, and so it's quite the opposite of an orderly, programmed process. And this is an idea we'll be coming back to throughout my talk here. But first, I want to introduce you to the grasses that I've been working with in the genus Brachypodium. And so you may have heard of Brachypodium distachyon. It's a diploid grass model for especially good for cereals such as wheat and barley and rice. And over here in the middle, we have the diploid Brachypodium stacyi, a lesser known species of the genus Brachypodium, and Brachypodium hybridum, which is the allotetraploid that was probably produced in nature many times from the hybridization of Brachypodium distachyon and Brachypodium stacyi. And what's really exciting about this model system is that our lab has actually just finished assembling the genomes of all three of these species. And we're just now getting this model system off the ground with some preliminary RNA-seq analysis. And that's what I'll be talking about in this talk. Additionally, we have another resource in the form of a synthetic polyploid that was formed in the laboratory by the hybridization of modern distachyon and stacyi to form this sterile F1 hybrid that was then made into a polyploid with this chemical called colchicine. And so together, these two synthetic and natural lines of Brachypodium hybridum form two different snapshots in the polyploidization response, the kind of immediate response and the more long-term response. So, as I mentioned, we did this RNA-seq experiment in these different genotypes, and I'm not going to go into my methodology, I'll just jump straight to the results. So the first question I had was, how many genes are differentially expressed in this polyploid relative to its progenitors? And the answer is about a third to a half, maybe, depending on which subgenome you're interested in. But I put progenitors in quotes here because I think it's important to keep in mind that these are not the true progenitors of the polyploid. The only way we could get those as if we had a time machine, but unfortunately, we don't have one of those, so we have to compare it to the modern diploids, and furthermore, to modern accessions that aren't necessarily the most closely related accessions to the true progenitors. So these numbers, a third and a half, are probably a bit inflated for that reason. But anyway, we can still contrast these with the number of genes differentially expressed in the synthetic polyploid, which is closer to around 70%. And in this case, of course, we're comparing it to the true progenitors, the modern diploids. So even in spite of the fact that the natural polyploid numbers are inflated, they're still lower than we see in the synthetic polyploid. Next, I was curious about the types of genes that were differentially expressed in each experiment. So I took the two lists of differentially expressed genes from these two experiments, and I compared them to each other, asking, are there any functional categories that are misexpressed in one polyploid but not the other? And that's how I produced this figure. And in particular, I'd like to draw your attention to this little cluster right here, which shows that functions involved in epigenetic gene silencing appear to be misexpressed in the synthetic polyploid, but not so much in the natural polyploid, which was encouraging to see because we know from the literature that synthetic polyploids often undergo a lot of epigenetic reprogramming as well as chromatin restructuring. So that is one aspect of the genome shock response that is somewhat predictable. And it shows that even though it's a huge number of genes that are misregulated in the synthetic polyploid, it's not just a completely random mess. There actually is a functional basis for this, and the genes that are being misregulated are not at least entirely random. So to summarize, gene misregulation is rampant, especially in the synthetic polyploid, and among the genes more frequently misregulated in the synthetic polyploid are those that encode epigenetic housekeeping functions. From these data, we can form a couple hypotheses. First, the shock of polyploidy involves large-scale transcriptomic and epigenetic responses. Well, that's not really debated much, but we can take that one step further and suggest that over time the shock wears off and the genome recovers, returning to a near parental state. So the number of genes that is differentially expressed goes down, and these epigenetic housekeeping functions are no longer so misregulated. But of course, there are still some genes that are very differently expressed between the progenitor and the polyploid. And so what about those genes? Are they randomly misregulated, or is there a good reason for it? And so to investigate this question, I focused on silenced genes, which are of course a very extreme form of gene expression misregulation. So the first thing I noticed was a very small number of genes are silenced in each polyploid, only a few hundred out of a genome of around 30,000 genes. And also that there were no genes that were silenced in both the natural and synthetic lines. So either the number of genes, either the pool of genes that's silenced is silenced completely randomly, or the two pools of genes are silenced for different reasons. So I dug a little deeper, and I found that in the natural line, gene silencing tends to occur in genes that are poorly conserved, both within and between lineages. And I'll just show you the data from within a lineage. So some other folks in my lab, for a completely different project, actually went around the world collecting ecotypes of the grass Brachypodium distachyon and sequenced the genomes of all these different diverse lines and categorized the different genes they found based on how widely conserved they were within the lineage. So core genes are those that are found in all ecotypes of Brachypodium distachyon, whereas soft core genes are less commonly found, and shell genes are unique to only certain ecotypes but absent from others. And so what I found when I looked at the categories associated with my silence genes was that they tended to be associated with the shell category. These are genes that are putatively involved in local adaptation. They're not required for survival. And these are the genes that get silenced in the natural polyploid. Meanwhile, in the synthetic polyploid, this trend is absent. So we don't see any sort of enrichment for these more dispensable, perhaps, genes. And so the conclusion that I draw from this is maybe the forces driving gene silencing in the natural polyploid are different from those driving gene silencing in the synthetic polyploid. Which brings me to my final slide and the idea that we may be able to start sketching out the trends in a polyploidy response. So initially, gene expression change is driven by these more kind of housekeeping factors related to genome structure and maintenance. Then over time, the polyploid largely returns to a more parental state, and the forces driving gene expression change shift to being more about demography and selection and these evolutionary forces rather than molecular ones. And so that's a topic I'm excited to pursue for my PhD, and I wanted to say thank you for your attention and for your support.